So now I'd like to introduce our two speakers for today's presentation. Mr. Jim Bruce is a Senior Product Manager at Life Technologies and is responsible for the MicroSEQ Microbial Identification System and other products related to the pharmaceutical QC market. Prior to joining Life Technologies, Mr. Bruce was previously employed as a Senior Account Manager with Molecular Epidemiology Incorporated and at Acugenics, a microbial service identification lab. As a research microbiologist, he was a member of the DuPont Qualicon Company, where he was responsible for the development of riboprinter microbial characterization system, and he also held the position of Senior Technical Account Manager. Mr. Bruce was also employed by the University of Pennsylvania as a Senior Research Microbiologist with the Large Animal Internal Medicine Group. Mr. Bruce has a bachelor's degree in medical technology from Franklin College of Arts and Sciences and also graduate coursework in microbiology at West Chester University in Pennsylvania. Our second speaker, Ms. Pia Darker, is currently the Field Application Specialist Manager for the Quality and Safety Testing of the Applied Markets Division for the Europe, Middle East, and Africa regions. Prior to her current role, she was deeply involved in helping to implement rapid molecular methods for different analytical methods in bioprocess and pharmaceutical microbiological quality control for different European and Asia-Pacific pharmaceuticals. She has extensive experience in the implementation of genotypic microbial IDs at well-known pharmaceuticals and also implementation of analytical methods such as mycoplasma and residual DNA testing. Ms. Darker has a bachelor's degree in microbiology from the University of Santo Tomas in the Philippines and a master's degree in molecular immunology from National University of Singapore. Ladies and gentlemen, it is my pleasure to introduce to you our first speaker, Mr. Jim Bruce.
Thank you, Tony, and I'd like to thank everyone for taking time out to join us on our webinar to hear more about the MicroSEQ Rapid Microbial Identification System. Before going into the MicroSEQ system, I'd like to give you a little bit of background on Life Technologies. At Life Technologies, we're very excited. We actually have been very focused on the areas of molecular methods, really focused around the PCR sequencing systems and reagents, as well as cell isolation and manipulation. And we've taken those methods, and we've been emerging over time into the areas in commercial, such as bioproduction and forensics, as well as clinical research in the areas of molecular diagnostics and genome-wide medicine. Basically, what we've done is we've taken these core competencies that we've had. We take in the areas of bioinformatics, in the areas of instrumentation, in the areas of reagents, and basically have developed total solutions, tools, and provided to these other applied markets. As an example, in the areas of forensics, we are now the leading manufacturer of forensic systems worldwide. And more recently, with the bioproduction, we've been focused on areas of quality control testing and contaminant analysis. The next is the areas, the focus of the business unit that we're involved with. And it's really, our focus is in product safety and product quality. And the business unit is called Pharma Analytics. Here we actually develop total solutions for the areas of process development, impurity analysis, and specifically in the area of QC microbiology, which we'll talk a little bit about today. So these are the two key platform areas that we work with. We work with real-time PCR systems. In the areas of product quality, we've developed assays or total solutions for mycoplasma, residual DNA, viral detection, and in the areas of product safety, microbial identification and detection methods. So today we're going to talk about the MicroSEQ ID system, which is a system that's focused on the identification of bacteria and fungi. So the question comes up is, what is the importance about microbial identification? And this is specifically important in the areas of bioproduction or pharmaceutical manufacturing, in the areas of the product manufacturing. As we all know, harmful bacteria and fungi can contaminate raw materials, production facilities, as well as the branded products out there. And we also know that in order to alleviate or at least to reduce that incidence of these occurring, the regulatory expectation is to include an active environmental monitoring program as part of their CGMP process. More recently, the U.S., European, Japanese Pharmacopoeia guidelines have started recommending the use of genotypic methods. The reason they are recommending these are because of the high level of accuracy that these methods provide. And in 2004, just to point out, here's an example of the FDA where they make mention that the genotypic methods have been shown to be more accurate and precise than traditional biochemical and phenotypical techniques. And these methods are especially valuable for the investigation of failures such as sterility tests and media field contamination as well. So the MicroSEQ Microbial Identification System, and in this case we've now come out with the MicroSEQ Rapid Microbial Identification System, is a next-generation high-throughput comparative DNA sequencing system for both bacteria and fungal identification. The system itself is an integrated system which includes bacterial kits, and we have two types of bacterial kits. One is our bacterial 500 16S ID kit. This particular kit amplifies the first 500 nucleotides of the 16S gene, while we have a full gene kit which will allow you to amplify the complete 16S gene. In the areas of yeast and molds, we have a fungal kit where we amplify the D2 region of the large subunit of yeast and molds. The system itself also includes, there's a 3500 and a 3500 XL genetic analyzer. These are different, really it's for different throughputs, as well as the MicroSEQ ID software, validated libraries for bacteria and fungal. And lastly, there's a thermocycler that is also part of that system. So the technology behind MicroSEQ, it's really, MicroSEQ is a phylogenetic approach. The MicroSEQ Microbial Identification System is based on 16S DNA sequencing. It's also, for fungi, it's based on D2 DNA sequencing. And this is, the reason we focus on this area is they are universal among bacteria and fungi. They're functionally conserved. They provide a high level of discrimination. And even more important, they are stable. They're not influenced by environmental conditions. So when you look at organisms that are grown in different environments, there is no impact on the identification. And are also, more recently, these are really a requirement for describing new species. So for us, we talk about going molecular, going from the phenotypic to the genotypic methods, and really the answer is in the DNA. As an example, if you go back to the evolution of DNA sequencing, back in 1965, it was first noted, the ribosomal RNA gene was first noted by Dubineau et al. In 1985, where we started seeing a lot of publications coming out, and this is just a reference to Woese and all, but there's many publications that came out talking about the phylogenetic relatedness based on the 16S gene. And then really in 1998 was the first recommendation by a pharmaceutical regulatory body that recommended the use of genotypic methods. And in the year 2000, Burgess, which we all know as a reference manual for microbiology, really had revamped or reorganized the structure of this classification, and it was all based on the DNA and as well as the 16S ribosomal RNA gene. And since then, 16S sequencing has really emerged into being a gold standard technology for bacterial identification. And as I mentioned, there's several guidelines that have coming out. In 2004, we heard from the U.S. FDA, 2007, the Japan Pharmacopoeia, and in 2008, the European Pharmacopoeia. They've all added recommendations of the use of either nucleic acid techniques or DNA sequencing as a way to identify microorganisms. So we'll talk a little bit about MicroSEQ ID and the emergence of MicroSEQ ID. So in 1998, MicroSEQ ID was first released as a research tool. This really, the focus of this particular product at that time, it was really use of kits and software which allows systematic microbiologists or bacteriologists to utilize this as a tool to describe new organisms or at least classify the microorganisms they were examining. In 2003 was the first attempt to go into an applied market with MicroSEQ ID, where we released a software version which was based on a PC with some 21 CFR Part 11 tools for the pharmaceutical industry. But in 2006 is when there really was that fully launched the integrated MicroSEQ microbial identification system. In addition, we also launched the new chemistry for our kits, which basically allowed us to reduce the time to results from 10 hours down to 5 hours, as well as a new software package which really addressed the key needs for the tools for 21 CFR Part 11 compliance. 2007 through 2008 and 2009, in recognizing the regulated markets, we had become focused by providing tools that would assist our regulated customers. As an example, in 2007, we released our IQ/OQ service. We've also, in 2008 and 2009, became more of a sole supplier of not only the MicroSEQ microbial identification system, but some of the other components that they may need to run DNA sequencing. And we're very excited by 2010. This year, we're very excited about the release of the MicroSEQ Rapid Microbial Identification System. And as I mentioned previously, this really involves a next-generation DNA sequencer with a higher throughput. So basically, since 1998, we really have been enhancing our products to meet the need of the market. So we'll talk a little bit about the key features and the benefits of the MicroSEQ Rapid Microbial Identification System. We are using the next generation genetic analyzer. It does provide first-time accuracy. And again, this is based on the ribosomal RNA genes. We're looking at that. We're doing amplification and getting high level of accuracy for identifications. And we do that through the extensive validated libraries that we supply with this system itself. We provide actionable results in less than 5 hours. Again, it's a 5 easy-step workflow. The software itself is very intuitive, and it contains autoanalysis, which allows you to load your samples and walk away and come back and review the reports that it's generated. It's a measurable financial impact. And the key things here is it reduces retesting. As you're all aware, if you're running some systems, you may have to go back and retest those organisms just to generate an identification for those particular organisms. With the MicroSEQ and 16S sequencing, it allows you to get that high level of accuracy, thus reducing the retesting that may be required. In addition, it reduces that contract lab cost. So for those who, if they do not generate a identification or have a confused identification, what I'm saying is a mixed results, then they will send them out to contract labs. This, by having a high level of accuracy, will reduce the need to do so. And another area that we've been focusing on, and that's in the area of validation. Those in the pharmaceutical quality control laboratories, we know the needs for validation. We know that can take a long time. So we really have reduced this through our IQ/OQ programs as well as our implementation programs. This allows you to really purchase and go through the process of validating your system in approximately 4 months' time, and allows you to utilize your system and as a routine identification tool after 4 months. And it's powered by the state-of-the-art genetic analyzer. These genetic analyzers are higher throughput than our previous MicroSEQ identification system. They now provide a plug-and-play consumables, which really makes it easy to use. And then it has the maintenance, scheduling, and tracking capabilities. So overall, the solution is going to the next step to meet the needs for ease of use. So some of the other features in a little bit more detail around the software. So the MicroSEQ, the software for the system, some of the key little tools that it has like automated reagent check, and this basically will reduce waste on your system. You have easy manageable maintenance schedules. It provides an autoanalysis manager and has improved features to support the 21 CFR Part 11 compliancy. And it is compatible with 3130 and 3130 XL genetic analyzers. Some of the key benefits, there is definitely reduced setup time over our past system. We have reduced maintenance and downtime, and ease of use in data retrieval and analysis, as well as efficient approval process for sign off your reports. And overall, it does provide better informational control. And this is a snapshot of the intuitive user interface. And what we're showing you here is a dashboard. And this is the dashboard really showing you the usage of your consumables. As you can see, there's different scales for the different consumables that are used on the instrument, but it really reduces the waste. And by having this, you're not only reducing waste, but you can also create your maintenance schedule, and the maintenance schedule will be reminded and also document when the maintenance is completed on your instrument. Another great benefit of the MicroSEQ Rapid Microbial Identification System is the increased throughput. So our past system was at 4 and 16 capillaries, and now we've moved into 8 and 24 capillary systems. The throughput is faster. You have a higher volume over a 24-hour period. As an example, in 8 hours for the 3500, you will get 16 identifications, and in 24 hours you can get 52 identifications. Whereas with the new higher throughput instrument, the 3500 XL, with 24 capillaries, you'll be able to identify within 8 hours 52 samples and 156 samples in a 24-hour period. So in general, the MicroSEQ identification system, the workflow itself is 5 easy steps, but the key thing here is it will provide a identification from an isolated colony in less than 5 hours. And what that means by that is if you have a primary plate with multiple colony populations, long as those colonies are isolated, you can actually take a small sample from each one of those colonies and generate an ID. So as we talk about in more detail about the actual process of the MicroSEQ identification system and the technology behind it, I'd like to take this time to turn this over to my colleague, Pia Darker, who will go through each step.
Thank you, Jim. So as Jim has mentioned, the MicroSEQ Microbial ID system is a 5 easy steps for you to get until you get your microbial identification. So it will start with growing a pure culture. So from this pure culture, all you need to have is a very small amount of cells that are needed in order to extract the DNA from these cells. The advantage of using a microbial ID system or MicroSEQ microbial ID system is that you don't need to have a prior knowledge of the organism. So you don't need to do a gram staining. As Jim have mentioned, because we are using a functionally conserved region as well, and because it is based on DNA, there's no need for it to be a fresh culture. And also, it's really ideal for slow-growing microorganism or stress microorganism, which is basically what you normally found in a pharmaceutical environment. So the first step is to have a pure culture. After that, once you get your pure culture, we can then do a DNA extraction. So during the DNA extraction, the idea is to get the DNA out from the cells. So again, we're using a reagent called Prepman Ultra. So this reagent is, we will add a loopful of cells to 100 microliter of this reagent Prepman Ultra. And all we need to do is to boil and lyse the cell. So we boil it at 100 degrees Celsius for 10 minutes, vortex it, and then we spin it down so that we pellet out the cell debris. And the supernatant is basically your DNA solution. We then dilute that supernatant into 1:100, and we'll use 15 microliter of the diluted supernatant for our next step, which is the PCR reaction, which has a total volume of 30 microliter. So with the next step, so after we got our extracted DNA, the next step is to amplify or to make a lot of copies of the 16S gene for bacteria or the D2 gene for fungal. So we are using specific primer sets that are specific to a conserved region in this target gene. So we're using a PCR master mix, which is part of the MicroSEQ PCR kit. So the primers are included already in this master mix with all the necessary reagents or components for amplification using the Verity thermal cycler as our PCR machine. So in this step, what it is that it is just a cycle of heating and cooling step wherein your primers bind to the sequence in your DNA, in the bacterial DNA or fungal DNA, and then it is just a cycle of denaturation, annealing, and extension of this target gene. So in the end, what we'll have is a lot of copies of 16S gene or the D2 gene. Because of the use of the PCR master mix, which is in a kit format, there is a minimal hands-on time. And as I've said, the master mix is ready to use. So these are all pre-optimized already. So once we have the amplified DNA, which is the target DNA, which is 16S for bacteria or D2 for fungal, the next step is to label this amplified DNA. So labeling is basically we're using another component or another kit, which is called the MicroSEQ sequencing kit. In this kit, it contains some fluorescently labeled nucleotides. So these fluorescently labeled nucleotides will then be bind into your DNA strand, either of the forward or the reverse strand. So we will have two reactions per sample. So one is the forward sequence and the other one is the reverse sequence. As you know, that DNA are complementary sequences. So during the sequencing reaction, these fluorescently labeled DNTP will bind to the DNA and will generate sequencing products. So after we generate the sequencing products, we'll then put the sequencing product into the automated DNA sequencer. So it will read the sequence. So in this sequencer, which is the 3500 or the 3500 XL, it's fully automated. So everything will be done for you from the polymer filling, sample handling, electrophoresis, and up to the data analysis. So once the raw data has been collected by the sequencer is connected to a computer system, it will then allow you to import the raw data sequences into the downstream software, which is called the MicroSEQ ID software. So the MicroSEQ ID software will then give the identification of the organism by comparing the sequence of your unknown isolate, which was generated using the sequencer, comparing that sequence to the validated sequences that is in the validated library, which is part of the MicroSEQ ID software packages. So the software package contains the analysis software and the 3 validated libraries. One for the 500, first 500 gene of the 16S. The second library is the 16S full gene sequence. And the third library is the library for fungal and yeast. So once the sequence has been compared to the database, it will then give you the match, the microbial identification match. So the next slide will just give you an example of what you see in the MicroSEQ ID analysis software. So one of the advantages of using a genotypic method for microbial identification is that it gives you a raw data. So in this slide, as you can see, this is an example of a raw data sequence, which is shown as an electropherogram. So by looking at this, it gives you an added confidence that the raw data of your isolate is really good, so that it gives you confidence that whatever the microbial identification that was given as a result is 100% accurate. So if you have a bad sequence quality, then you cannot trust really the identification result because of a bad raw data. So the next slide will give you an example of how the software reports the top 5 matches. So as you could see in this slide, there are top 5 matches for that particular isolate. It gives you a % match. Obviously, the one that with the highest match is the right or the closest identification for that particular isolate. And also, it gives you a phylogenetic tree. So it gives you a more kind of a representation, an evolutionary representation of the relationship of these top matches for that particular isolate. As I mentioned earlier on, the MicroSEQ microbial ID system comes with a validated library. This is the most important part of the system because this is where the sequences of your unknown isolates are being compared to. So the sequences in these validated libraries are fully validated, which means that we have really done an extensive validation to make sure that the sequences are correct. So for the 16S500, we have more than 2,000 entries. For the 16S full gene, there are more or less around more than 1,200 entries. For the D2 or the fungal database, we have more than 1,100 entries. The libraries are being upgraded or updated regularly, especially with new entries. Mostly are pharmaceutical or environmental entries, and also we update it with the updated nomenclature. As you know, there are a lot of reclassification going on because of the fact that now the Burgess manual is now using 16S as a gold standard for taxonomy. So there are a lot of changes happening with the old nomenclature for bacterial isolates. So as you can see that even though we have an extensive number of entries in our library, obviously these are not enough, as there are a lot of microorganisms available in our environment. So if you have some species that are not in our library, you can actually suggest some entries to be put into the next update of the library to our website, www.microseq.com. So the MicroSEQ Microbial ID software validated libraries is so far the largest, most comprehensive validated libraries for bacteria and fungal available commercially. So with the system, you can also we also have some validation certificates that are available for you to be downloaded to our website, or you can request it from your local sales representative or application support representative. So these validation certificates basically highlights to you the steps we do on how we validated each entry or each sequence entry in our libraries. It also comes with software certificates for the MicroSEQ ID software as well. So as Jim has mentioned, the system comes with we offer some qualification services like the IQ/OQ, as we know that this is a requirement for a regulated environment such as the pharmaceutical quality control. So it comes with an IQ/OQ qualification service. So our service engineer will provide this installation qualification and operational qualification as part of the implementation of the system. Also, it comes with an implementation program. So this implementation program, the user or of the laboratory. So the 3-part training that are divided into 3 parts, depending on the knowledge or the experience of the user or of the laboratory. So the 3 parts of the training can be divided into initial training, which means that it's just basically a discussion or giving a basic molecular biology information to get familiar with the technique of PCR sequencing and the DNA as a technology for microbial identification. Then is what we call the workflow training. In the workflow training, we discuss more of the protocol or the process from sample preparation down to data analysis. It's more on hands-on training. It discusses the maintenance of the instrument and some basic troubleshooting. The last phase of the training is what we call the completion training, which is basically a discussion of the experience during the performance qualification and also some advanced troubleshooting as well. So also during this implementation program, we also have some performance qualification guidelines that you can use to give you an idea on what you could do as a performance qualification protocol when you do your PQ for the MicroSEQ system. So all these information about the IQOQ and about the implementation program can be found in our website, www.microseq.com. So the next few slides, I just would like to give you some case study. So it's a general case study of what we've done or the experience of most of our customers after the implementation of the MicroSEQ ID system. So the first case study is basically an experience of a global manufacturer of pharmaceutical preparations, wherein they decided to change their phenotypic identification into a genotypic identification. So before MicroSEQ ID, what they have found is that they have a lot of inconsistent results, meaning they have a lot of repeat testing, and often require them to actually outsource some of their critical isolates for genotypic identification, because there's a requirement by CGMP that for critical isolates or isolates coming from critical sterile areas, it needs to have an identification down to species level. So they normally outsource this identification because they do not have a genotypic method in their facility. So normally they only identified more than 90% of the organisms in the plant have only one or two species identification. So after MicroSEQ ID, what they have experienced is that they have virtually no repeat testing necessary, and they have a much rapid, accurate result that streamline their product release process. So basically, because they are a support, this QC lab is a support for their manufacturing facility, so they can actually release the result in a shorter period of time. So the next case study that we have is from a global manufacturer of vaccines, wherein they've used a genotypic method, or they required a genotypic method for a root cause investigation. So before the MicroSEQ ID, the manufacturing license of this particular company has been suspended due to contamination and sterility issue in manufacturing facilities, wherein they failed to actually investigate the root cause of the problem due to misidentification. After the implementation of MicroSEQ ID, they were able to have a rapid, accurate ID, which helped them to put some remediation efforts or preventative actions. This has been now incorporated in their new processes, and it actually assists by implementing MicroSEQ ID system. It actually assists in the earlier reinstatement of their manufacturing license. So with these two case studies, it's just to show you that the use of the MicroSEQ ID system in the pharmaceutical environment has been increasing in the past 3 to 4 years. And as a result of this successful implementation, 10 of the top 10 pharmaceuticals worldwide has been now using MicroSEQ identification system. 21 of the top 25 are actually MicroSEQ ID users, and there are 15 multi-site adoption in the top 25 pharma that now basically implemented MicroSEQ ID system in their sites worldwide. So to summarize, the MicroSEQ Rapid Microbial ID systems gives a high accuracy identification and gives quicker results, basically quick rapid time to results, easy to use. So with a very intuitive software with autoanalysis, makes the workflow streamlined. Simple 5-step workflow. It reduces your daily setup time and also make the maintenance procedure easily with the system dashboard system of the data collection software. Fast installation and validation time with the help of the qualification services that comes with the system and implementation program. And with the improved throughput of the 3500 analyzer, you can have either 8 or 24 capillary system, which comes with pre-packed consumables, which enables you to have an easy maintenance of your reagents and consumables. So with that, I would like to thank you for listening, and we would like to answer your questions.
Thank you, Pia. Thank you, Jim. It's time to open up the floor for questions. So if you have a question, please submit them via the chat function on the right-hand side of your screen. We've posted some instructions. And give me a few seconds, and I'll read the first question to everyone. First question, does the system require specific culture media for identification?
No, actually, the nice thing about the MicroSEQ system and its DNA sequencing does not require any specific media, and it really, the media itself will not influence any of the results on the system for the identification or DNA sequence.
Next question. Does it require fresh culture?
I'll defer that one over to Pia.
Pia, does the system require fresh culture?
Oh, no, the system doesn't require fresh culture. As I mentioned earlier on, because it is based on DNA, which is a conserved region, and it's not affected by environmental conditions, so we don't really need it to be a fresh culture. So even though you have a week-old culture or a month-old culture, the system can still identify that particular organism.
Great. Next question. Is the system FDA approved?
No, it's a very good question, Tony. No, the system is not 510K approved. However, in the markets that it's utilized, that's not a requirement for the use of the system. So as an example, the system is validatable. So many of our customers, pharmaceutical customers who are in the QC laboratories have implemented this and validated under the requirements by the regulatory agencies, and then have passed their requirements and adopted and implement MicroSEQ ID for testing.
Next question. How frequently has the library been updated?
That's another good question. And we try to update it on a regular basis. Basically, our rate is about once every 2 years. We are now re-looking at our program, doing some modifications. Our goal is to do it more frequently than that, but it currently has been once every 2 years. And again, our focus is to knowing that the changes in nomenclature happen quite readily, and also the needs to identify newer organisms that are either not classified yet or new species that have been described. We're going to have a program that will be much more routine release of updated libraries.
Are there problems with IDs of closely related organisms?
And I'm going to defer that one over to Pia. Pia, could you answer that question?
Yeah, sure. Yes, because the fact that sometimes the first 500 base pairs of the 16S, there are some very closely related species. So if there are some closely related species, then what we recommend is that to run it using the full gene kit. Normally the full gene kit will then help you to basically differentiate very closely related species. Now, now there are some species that are really close, even though in a full gene kit, we have some guidelines that will help you to go through to get a final identification. Actually, it is part of our data interpretation guideline, which discusses on how to handle this type of identification results.
Give me 1 second here while I prepare the next question. Is the software updated with each update of the library?
No, actually, our software upgrades and libraries are kept separate. When we update the software, although we like to add a new library at that time, a lot of times it might be a minor upgrade for the software, so we've kept them separate for that particular case.
Great. I'd like to let the audience know we don't have any further questions. If you have any other questions, I'll stay on the line for another 5 to 10 seconds. If anybody has any other comments or questions they'd like to ask. Okay, with that, I'd like to thank everybody and appreciate everyone for participating in this seminar. If you've enjoyed this seminar and would like to learn more about the Bioproduction webinar series, please check out www.lifetech/bioprocessing, or you can email us at bp@lifetech.com. There's also a link that you can see on your screen to the Meeting Bridge site where you can view our 2011 schedule and also previously recorded events. Each month, we'll be offering different presentations on cell culture development, downstream purification, and rapid molecular-based contaminant testing. Our next webinar is scheduled to take place in late January, and we hope that you join us again for one of our future webinars. Thank you again, and